Good morning. This video is of lesson 10, that is cell cycle and cell division continued. We have studied till mitosis. After mitosis, the next stage of cell division is meiosis. Now, in meiosis, there are two M stages, that is, one is M1 and another is M2. In M1, we have prophase 1, metaphase 1, anaphase 1, telophase 1, and in M2 we have prophase second, metaphase second, anaphase second, and telophase second. Meiotic prophase 1 is of long duration as compared to mitotic prophase. So what happens in meiotic prophase में prophase further 5 stages में divided है. उन stages का नाम है लैप्टोटीन, zygote, पैकेटीन, diplotene, and dikinesis. Now, lactotine. What happens in lactotine? In lactotine stage, there is differentiation of genetic material into chromosomes. The genetic material, which is in the form of network, has started distinguished into chromosomes, and the chromosomes become gradually visible under the light microscope, and they have started compacting. So the next stage is zygote. The word zygo means pairing. So zygote, in this stage, the pairing of homologous chromosomes take place. Homologous chromosomes are those chromosomes which in which one chromosome comes from maternal and other chromosome comes from father. So in zygote, the process of pairing of chromosome is called synapsis, and the paired chromosome bivalent is paired homologous chromosome is called bivalent. So micrograph of electron micrograph at this stage indicate that chromosome synapsis is accompanied by the formation of, there is another complex structure called synaptonemal complex. This complex formed by a pair of synapses homologous chromosome is called a bivalent or a tetrad state. The next stage is packeting stage. You know, in packeting what happens? There is exchange of genetic material between non-sister chromatids of a chromosome take place, and in this the point where exchange of crossing over take place, they are X-shaped, cross-shaped structure, and they are called chiasmata. The single one is called chiasmata and the plural is called chiasma. So that is why ये क्रॉसिंग ओवर की वजह से ही आप जो भी वेरिएशंस देखते हैं पेरेंट्स में, चिल्ड्रन में या उनके सिबलिंग्स में, तो दिस इज ओनली बिकॉज ऑफ क्रॉसिंग ओवर in case of sexual reproduction. So crossing over is also an enzyme mediated complex. इसमें enzyme की भी जरूरत होती है और जो enzyme इसमें participate करता है उसका नाम है recombinase. So this phenomena is also called recombination. That means new combination of genes. Now next is diplotene. In diplotene stage, a synaptonemal complex which was formed earlier has started dissolving now, and tendency of the recombinant homologous chromosome of the bivalent to separate from each other except at the site of chromosome, and the final stage of meiosis 1 is dikinesis. In this, what happens? Chiasmata, cross-shaped structures, they start moving towards the terminal site, that is terminalization, and other structures like disappearance of nuclear membrane, nucleolus, they start happening. Now, next is metaphase 1. Now we'll look at this diagram, and in this diagram, prophase 1, here the prophase 1, see how the chromosomes, these are the X-shaped structure, and they are the chiasmata, and there is disappearance of nuclear membrane and nucleolus. In metaphase 1, the chromosomes has shown maximum condensation and this is the stage where we can count the number of chromosomes. So in this, chromosomes, they get arranged in the center towards the equatorial plate, and in this anaphase, the chromosome in prophase, in mitosis stage, instead of chromosomes separate, chromatid separate, but in this case the chromosomes, they separated, they start moving towards the poles, and at the end of the anaphase 1, that is telophase 1, two nuclear groups with half of the number of chromosomes. Earlier we have 4 chromosomes, but in these daughter cells we have 2 chromosomes. So this is M1 is reductional division, and now second is M2. M2 is called equational division. In this M2, what happens in prophase second, again the disappearance of nuclear membrane, nucleolus, they start disappearing. In metaphase second, chromosomes, they have started getting arranged themselves towards the equatorial, and spindle formation is there. And in anaphase second, again we find two separate nuclear groups. So the result of M2, that is after anaphase 2 and cytokinesis, we are getting 4 daughter cells. Earlier, from 2 parental cells, we are getting 1, 2, 3, 4 daughter cells with half of the number of chromosomes. So meiosis second is similar to mitosis. So next topic is significance of meiosis. You know, meiosis is very important. It maintains the chromosome number constant. If there is no meiosis, then the next new organism formed may be tetraploid, may be hexaploid. So meiosis results in the formation of haploid gametes, and haploid gametes form the diploid organism, and thus the chromosome number of a particular species is maintained. Another significance of variation is that it brings variations, and variations are important raw material for evolution. So new species is evolved through this genetic variability which occurs in meiosis second. So today we have studied meiosis M1, M2 and significance of meiosis. Thank you.